Okay, so since there were so many of you that were out or had National History Day, I wanted to go over the notes that we did in class on Tuesday, February 4th, 2025, so that anybody who wants it can have access to them, even if you were in class when we went over these things. It's just another way that you can go over it in preparation for your test next week. So, like I said in class, Mendel was really good at figuring stuff out in genetics, but he kind of stuck to the idea that for all traits, there's a dominant trait, a dominant form of that, and a recessive form. Dominant one's always going to cover up the recessive allele. Recessive can be hidden for generations and then pop up later. But there are some traits that don't follow that pattern, that are what are called non-Mendelian traits, where there's a little bit more going on. As a reminder, we have 23 pairs of chromosomes. This picture of a chromosome or a person's chromosomes all laid out like this is called a karyotype. So if we're looking at these 22 pairs of homologous or 23 pairs of homologous chromosomes, the first 22 of them are what are called autosomal, which means that they are not sex-determining chromosomes. That last pair is the sex chromosomes, which are going to determine what kind of sexual development you go through. So if you are somebody who has an X chromosome and a Y chromosome, you are going to have phenotypically male characteristics, versus if you have two X chromosomes, you would have female phenotypic characteristics. As we talked about in our meiosis and non-disjunction kind of conversation, there can be some situations where you have different numbers of chromosomes besides this 23 pairs or 46 chromosomes total. And that can happen where you have too few chromosomes or fewer chromosomes, or sometimes more chromosomes for certain ones of our homologous sets. So when we're thinking about how traits are inherited, it could be inherited on one of those autosomes, those first 22 pairs, or it could be inherited on that last pair of chromosomes on the sex chromosomes. When we're comparing the X and Y chromosomes, if you're looking up in this top right-hand picture, the one on the left is the X chromosome, and the one on the right is the Y chromosome. You can see that the Y chromosome is a lot smaller than the X chromosome. The Y chromosome only holds a very small portion of the information that you would find on the X chromosome. So there are some traits that are found on the X chromosome, the genes for them are found on the X chromosome, but missing from the Y chromosome. So if you are somebody who has X and Y, instead of having two genes for that trait in your genotype, you would only have one. These can be dominant or recessive. Typically, when we're looking at X-linked traits in the first year biology, we look at recessive traits, X-linked recessive. These X-linked recessive traits are going to be much more commonly seen in men. The reason for that is because if you're looking at these different possible genotypes, if you're a female, you could have three different genotypes that are possible. You could have a dominant allele on both of your X chromosomes. You could have a dominant allele and a recessive allele on your two X chromosomes, or you could have two copies of the recessive allele on your two X chromosomes. What that means is for those first two, where you're homozygous dominant or you're heterozygous, you would have that dominant phenotype. The only way that you could have a recessive phenotype and show that trait is if you inherited two copies of that recessive allele. One on the X chromosome you got from your mom, one on the X chromosome one on the X chromosome that you got from your dad. However, if you are a male, there are only two possible genotypes. You either have the dominant allele on your X chromosome and you're missing the gene entirely on your Y. In that case, you have the dominant phenotype, or you have the recessive allele on your X chromosome that you got from your mom, and your dad contributed the Y, which again doesn't have anything in it. So for males, you either have it or you don't. There's no way for a recessive trait to be covered up by a dominant allele. So you couldn't have male carriers in X-linked traits. You either have it or you don't. There's no heterozygous option for you, which is why we see these recessive traits more frequently in males than we do in females, because the females have that other X chromosome that is potentially going to have a dominant allele and cover it up if they are also carrying a recessive allele. So when we're looking at this pedigree to the right, this is the royal family in Europe that came from Victoria and Albert and their kids, which were monarchs all over the place in Europe and in Russia. we're looking at the trait of hemophilia. This pedigree does show us the carriers. You can see that we've got a lot of female carriers. There are no male carriers because you can't be a carrier. You can't have the recessive allele and not show it for an X-linked recessive trait if you are a male. The only people who actually have the disorder in here are males. That's not to say that females can never have X-linked recessive traits. It's just in this particular case, none of the females married men who also had had hemophilia to pass on an X chromosome with that recessive allele. Typically, the reason for that, this kind of scenario where you're not seeing those males breed or pass on their genes, is because they don't live that long. And so, if they don't live long enough to pass on their alleles, they're not going to have affected kids because they didn't have kids. It is possible for females to have X-linked traits, but they have to have a father who was affected by that trait in order for them to inherit an X chromosome with a copy of the recessive allele from both mom and dad. So you can see them, it's just not as common to see females with X-linked recessive traits. Okay, so for dad can either give the X chromosome or the Y chromosome in his sperm. If he is carrying, if it is a female, he is contributing his X chromosome. If the kid that they have is male, he's contributing the Y. The eggs have two X's. Eggs are always going to have an X chromosome that goes into their offspring. So if you have an X chromosome, you can thank your mom for it. If you have two of them, you can thank your dad also. All right. On your packet that I gave you that looks like this, it says heredity and genetic crosses on top. The practice problem number 9, it deals with that concept of X-linked recessive traits. I'm going to have posted the key for that, so if you want to check and see your work, if you weren't here for when we did that, you can go and look at that key. All right, we've done pedigrees before. This allows us to track traits throughout generations in a family. It enables us to identify what's called the mode of inheritance. What I mean by mode of inheritance is either it's how is this passed? What's the pattern that it's following? Is this being passed on an autosome, one of your 22 pairs of chromosomes that is not a sex chromosome? Is it being passed on one of the sex chromosomes? So we have the first option is it autosomal or is it sex linked? Once we figure that out, then we can figure out, okay, is this thing dominant or recessive? So your modes of inheritance would be things like autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive. And we can figure that out based on the patterns that we see in pedigrees. When we're looking at this pedigree over here, this is a recessive trait. I can tell that because these two parents in F1, our F1 generation, have a kid, or have two kids rather, that are affected by this trait. The only way that we could have a child that is affected by this trait from two unaffected parents is if those parents were carrying it, if they were hiding that allele in their genotype and passed it on to their kids. Mom has to have a copy and dad has to have a copy so that their kids can get two copies on the pair of homologous chromosomes, so their genotype would be homozygous recessive. I can tell that this is not X-linked because remember, the only way for you to have a female that's affected by an X-linked trait is if her dad also had a recessive allele to pass to her. So since her dad does not have a recessive allele because he's not affected by it and he couldn't be a carrier of an X-linked trait, we know that this is an autosomal recessive disorder. If you see a dominant trait in a pedigree, generally it's going to show up every single generation. It doesn't really skip that much. With dominant traits, you can't be as definitive with your determination as you can be with a recessive allele, with a recessive trait rather. So with dominant ones, we look for probability of it being dominant rather than saying this is definitely a dominant allele, because it's a little harder to actually make that determination definitively. All right, so if I'm looking at this pedigree, the way that I would figure it out, the way that I would think about this of what's the mode of inheritance, is it autosomal, X-linked, dominant, recessive? I'm seeing it show up in every single generation, which indicates to me that this is probably going to be a dominant trait. I can't say with 100% certainty that this is dominant, but it's more likely to be dominant because it's showing up, it's not skipping. I would need more information to actually make a very definitive statement about that. As far as autosomal versus sex-linked, for that one, I'm gonna look to see, is this something that's more common in one sex or the other? Is this more common in males? Then I'm probably looking at an X-linked disorder. Again, with this one in particular, since it's dominant, we can't say for 100% certainty with this, but we can make a conclusion that we could support with some logic that this is probably an autosomal dominant disorder. It's showing up evenly in males and females. It's showing up in every single generation. This one would be a recessive X-linked disorder. It's not showing as carriers. When we were looking at the hemophilia one, where'd it go? I've lost it. When we were looking at hemophilia, there it is. This one did show us the carriers as those half-shaded ones. Not every pedigree is going to do that. Some pedigrees don't show you the carriers. It just, their phenotype is that they're not affected. So we might not know if they're a carrier or not from just looking at people, or you won't know from just looking at people. And so some pedigrees don't include that. So don't assume that if there are no half-shaded ones, then there's no carriers. This one has carriers, but they're not shaded in for us. On your test, I will not shade in carriers, because if there are carriers, that automatically tells you that that's a recessive trait. And I want you to be able to figure these things out. All right, so I'm looking at this one. This is a recessive trait for the same reason that our first example was a recessive trait. I have parents 1 and 2 in my P generation here, my first generation. They have kids that are affected. The only way for that to happen is if they are, if this is a recessive trait and they are carriers for this thing. That also tells me that when I'm looking at this is also a sex-linked trait. The reason that it is sex-linked is when I'm looking at who is affected, it's a bunch of males. We don't have any males with affected daughters to guarantee that this is an X-linked recessive trait, but we do have only males that are being affected by this trait, whatever it is. So this is an X-linked recessive disorder. It's skipping generations. I have unaffected parents who are having affected kids, and the affected people are all male. All right, this one is it's a portion of that first hemophilia pedigree that I showed you. This is still an X-linked recessive. This is one where we do have our carriers shaded in. So both of these are X-linked recessive. This one just shows me the carriers, and I only have female carriers, and only males are affected here. So this is an X-linked recessive. I have unaffected parents with affected kids, and the only carriers are females, and the only people affected are males. X-linked recessive. We can track a lot of inherited disorders by looking at single genes, so looking at a genotype for just one thing. And so pedigrees are really great for tracking these things and figuring out the likelihood of, if you're having kids and this thing runs in your family, what's the likelihood of passing that on to your children? You guys are doing projects on some of these things. Almost all of them are options for your genetics project, genetic disorders project. So you'll learn a little bit more about these on Thursday this week. Okay, our next non-Mendelian trait pattern of inheritance. We had X-linked traits or sex-linked traits. There are some Y-linked traits, but they're so few and far between that we just really focus on the X-linked ones because the Y just doesn't hold a lot of information besides your sex determination. Okay, so besides sex-linked traits, we also have some situations where you don't have one allele that is dominant to the other. There are a couple of scenarios where you can have some that are co-dominant, where both of them are dominant and they both show up. And sometimes they're incomplete in their dominance. They can't fully cover either of them up. Like the one can't cover up the other. Instead, you get a blending. So in incomplete dominance, my first one up here, the ones that are heterozygous, where you have an allele for one form of the gene, one phenotype, and you have an allele for the other one, instead of looking like either of them, they're going to blend. Neither one of them is dominant to the other. They just kind of become this one blended thing, an intermediate phenotype. So if I have a red flower and a blue flower, and this is an incomplete dominant scenario for that trait of color, the flowers that they would have as offspring would be purple. It would blend those two phenotypes. Sometimes, however, you have ones that one isn't dominant over the other, but they don't blend. Instead, they both show up separately. So if you have an allele for one trait and an allele for the other form of that gene, you're going to see both of them show up, but they're not going to mix. That is what's called codominant traits. They're both the same level of dominance, they both show up, but they're not blending together. If I had my same blue and red flowers, instead of having purple, which an incomplete dominance would be the case, instead I have a flower with some blue patches and some red patches. It's just both of them are showing up, but they're not mixing together. All right, on your packet again. This is question number 5. We did make some changes in class to that because sickle cell anemia is kind of a weird disorder. It's a weird trait that, depending on your circumstances, can show up in a couple of different inheritance patterns. It sometimes looks like a recessive disorder. It sometimes looks like a codominant disorder. So in sickle cell anemia, in this question, it says that it is heterozygous individuals would have a big A and a little a. If it's codominant, that's not a recessive allele for that second one, for the sickled proteins. Instead, it would be another uppercase letter. So the updated version of that question would read, in humans, the allele for sickle cell anemia exhibits codominance. Individuals who are homozygous for the wild type allele, meaning they have normal hemoglobin production, big A, do not have the disease. Heterozygous individuals with one wild type, big A, and one sickle allele, big S, because they're codominant, they're both uppercase, have a mild form of the disease. And homozygous sickle individuals, instead of homozygous recessive, they would have the genotype big S, big S. Homozygous sickle individuals have sickle cell anemia. In the Punnett square below, we have 2 individuals who are heterozygous for this trait. So they would have the genotypes big A, big S, and big A, big S. The reason for having a mild form of this disease, even though it's codominant instead of incomplete dominance, is because some of the blood cells, your red blood cells, the hemoglobin that they've made will be typical, and they will be the normal functional red blood cells. But some of your blood cells, instead of making the typical hemoglobin, are making this sickled form, this mutated form. And so those ones are not going to function. So rather than having all dysfunctional red blood cells like you would have if you were homozygous for sickles, you have some red blood cells that are functional and normal, round shaped. They do their job great. They have hemoglobin that works. They carry oxygen all around. Some of those are great, but then you also do have some sickled blood cells. So it is codominant because both of them show up, but they're not mixing. Generally speaking, people who are heterozygous for this trait, it's going to appear like they don't really have the disease unless the environmental conditions are a certain set of things. All right, so again, I'm going to post the key to this so you can check and see your answers and compare them to mine. Okay, in most classes, this is the last thing that we covered. I am going to keep going through the other things because I think that they are interesting and that they are worth knowing. But this is where we stop notes in B block and in A block. D block got a little bit further on that. So our next non-Mendelian thing, what we've had so far is sex-linked traits, incomplete dominance, codominance. Now we've got another one of multiple alleles. For some traits, rather than just having 2 options for forms of a gene, you can have more than those 2 options. It's not just you have a dominant one and another dominant one, or you have a dominant and a recessive. Sometimes there's more than 2 options for forms of genes. Blood typing is one of those. Your blood type, your phenotype would be, or your blood type can either be type A, type B, type AB, or type O. What that's referring to is proteins on the surface of your blood cells. It's a little hard to see on the slide here with the size that it is, but in the middle column here, we're seeing what the phenotype of that person is. What do their blood cells look like? So if you are somebody who has type A blood, what that means is your cells have these little proteins on the surface that they don't actually look like A's, but they are a form of a protein that was one of the ones we identified. We just gave it the designation of this is protein A. So if you are somebody with type A blood, your red blood cells are covered in A proteins. That's a way for your body to know that those are your cells, to recognize those as your cells. Any cells that have just A on them, you're cool. Is when your body cells, your immune system cells see something that is not normal, that they're not used to, that they would attack those blood cells. So if you're somebody with blood type A, you have A proteins on your blood. If you're somebody with blood type B, you have a different form of proteins that are on the surface of your red blood cells that your immune system recognizes. So if you have type B blood, your blood cells are covered in B proteins. If you are type AB, you inherited a copy of the A protein gene and the B protein gene. So those are going to be co-dominant. We have those ones that both show up. They are not blending together. They both show up. And so the blood cell has some A proteins on it and it has some B proteins on the surface of it. Our last blood type of type O is a third allele that codes for it. Type O blood has a form of the gene, the person who has type O blood has a form of the gene that does not code for any proteins, is a lack of protein. So your blood cells wouldn't have anything on them. You have a recessive allele that codes for that. So our options for alleles for blood type are big A, big B, and a little o. They have a recessive option. So we have two that are co-dominant, A and B, and then we have one that is recessive, O. So if you're somebody with type A blood, there are two genotypes that could give you that blood type. You could have an A and another A allele, which are both dominant alleles. You're homozygous dominant, or you could be heterozygous, where you have a big A and then you have that O recessive allele. It's recessive, so it gets covered up. We're going to make some A proteins, so we're not going to see a blood cell without any proteins on it. So that is A is dominant to O. So my blood type is A. I could be AA or AO. Same thing with type B. If my blood type is type B, it means that I have B proteins on the surface and that's it. So I could have BB or I could have BO, have a dominant B and that recessive O. If you have type AB, that tells you your genotype. You have the genes, you have the information to make A proteins and B proteins. That's your genotype. You have an A allele, you have a B allele. Our last option also only has one genotype because O is recessive, because if you had A's or B's, you would see them on the surface of your protein of your blood cells. If you have type O, you got two copies of that recessive allele. You have OO. This one is covered in practice problem number 8. All of the things that I just said are also explained in that, of the codominance and recessive nature of these different alleles. So you can look at that one and then compare it to the key that I will post. All right, so these are the ones that some of the classes didn't get to. Polygenic inheritance, just like you can have one gene that has more than one form of the gene, sometimes one trait has lots of genes that go into the determination of what you look like. So rather than, okay, I'm looking at my chromosomes, this one part, this one location that has the gene that has the information for this trait. Instead, if you're looking at a polygenic trait, you're going to have genes all over your genome in lots of different places on lots of different chromosomes that play into your overall phenotype. So it's not just one gene that determines your skin color, for example. You have many genes that go into determining your skin color. And so you can have a lot more options, a lot more variation in the overall phenotype, because if this set of genes is what codes for how much melanin you produce. If this is on this, it's basically saying that there are 6 alleles, or 6 genes rather, excuse me. There are 6 genes that go into determining your melanin production. If you inherit more of the no melanin production, you're going to have much lighter skin. So these ones up here got lots of no melanin production alleles. And then over here, depending on what combination of those alleles for the different genes you get, you may end up with more melanin producing genes. So you have all these genes in different places that are determining how much melanin did you produce overall. So it's not just one location, one gene that determines this one trait. You have many genes on lots of different places in your chromosomes that go into determining your one phenotype. Height is another one like this. You're not just either very tall or very short. We have all kinds of range of height in our human population. So these are ones that you'll see a spectrum of different options. All right, this is one that I think is cool, and because it's personal to me, I think it's really cool. Sometimes you have genes that control other genes that impacts whether or not another gene can actually be expressed. In dogs, specifically in retrievers, there is one gene that codes for if you can make pigment at all. And then there's another gene that says, what kind of pigment would you make? If you're making pigment, is it going to be black or is it going to be brown? So when I'm thinking about what color is my dog, first I need to know, does my dog make pigment? Can they make pigment? Period. If the answer to that is no, then you're going to have one of your yellow labs or your white labs or white retrievers. If the answer is yes, now we need to look at, okay, so what color are they making? So then, yes, they make pigment. Now we can figure out, is that pigment black or brown? If they couldn't make pigments, it doesn't matter what they have for this gene over here. They can't make it. They might have instructions, but some of the other machinery that's necessary for making those is missing. You don't have the ability to actually express that gene. So epistasis, you have one gene. What you have for that one determines if you can express another one. The reason I care about this is my dog's parents, Gunnar and Stella. Stella had a litter that looks like this. So even though we have we have a white golden and a chocolate lab, all of their puppies were little black puppies. This one's mine. His name is Moose. He's much bigger now. He's like 115 pounds now. So he doesn't look quite so compact anymore. But the reason that this is possible for these dogs to have a litter of all black dogs, even though it doesn't seem like that should be the right thing, is because of that epistasis. Because the most likely scenario is that this was our combination here. They all inherited the ability to express their pigment genes, and what they inherited for pigments was the dominant alleles. All right, this has come up when we're looking at your genetics projects already. Sometimes there are traits that are determined by if you had a major thing happen to your chromosomes. This is usually something that happens in crossing over in meiosis. So if you have not just like there's some random mutation because DNA replication or repair happened wrong. Here we have a major flaw with your chromosomes, a major difference from your chromosomes. An aberration is something that is out of the norm, unusual. So sometimes a big chunk gets deleted. Sometimes it gets repeated or duplicated. Sometimes it gets flipped or inverted. So all of these, the top one is what it usually looks like. And then if it has that aberration, the one underneath it is what that chromosome looks like instead. Sometimes you accidentally swap things that aren't homologous when crossing over happens. So sometimes there are mistakes where something that should be on one chromosome is actually totally elsewhere on a non-homologous chromosome because we had some errors in crossing over. That's called translocation. Lastly, how you express your genes is dependent in some cases on your environment and on your surroundings, so whatever that may be. That may be about the food that you're eating. It may be about the compounds that you're exposed to. It may be about temperature. So these examples here are ones that are temperature dependent. Our Siamese cats over here, their darker fur colors are places on their body that are cooler than other places. If those extremities get a little bit colder than the core. And so because of the temperature of those locations, the way that those cells express pigmentation changes. And so the temperature of those extremities leads to a different pigment expression than the rest of the body. Over on the right here, we've got turtles. Turtles' sex determination is not the same as ours. It's not like you have XX chromosomes, you're a female turtle, XY, you're a male turtle. Their sex determination happens in the egg before they hatch based on the temperature of their surroundings. If there are mostly warm temperatures while that egg is going through the developmental stages, it's going to be a female. If the temperatures were very cool, that's going to be a male turtle. This is something that's especially relevant when we think about things like climate change. If we've got things like sea turtles where they are low in number, they are endangered. If we have climate change and temperatures are going to be consistently higher, we're going to have a lot of female turtles and no males for them to reproduce with. So we have a different problem besides our like hunting and boat strikes and fishing and all of the other things that we've done that have impacted their population numbers. Now their ability to repopulate may be affected by temperature change that allows them to express their sex determination during development. This happens to you too. So it's important that you take care of your body, your height. If you don't get good food early on in life, if you have tall genes, too bad. You can't actually grow because you didn't have the nutrition for it. So, all of these characteristics here are things in you that you have some control over based on your environmental conditions if you develop in certain ways, if you get some of these different conditions. They are based on your actions and your ability to care for yourself. So, get lots of sleep, eat lots of food that is good for you, and relax. Try to not stress so much. All right. Hopefully, that was helpful. And now you got to spend even more time with me outside of school. You're welcome.